Welcome to another episode of Whose Gene Is It Anyway? I am your host, as always, Justin. Welcome to the show. Today's going to be a fun one, although hopefully shorter than usual. I have a lot of editing to do. Like, a lot. So, we're going to try and keep it concise today. As you may have seen from the title and thumbnail and all of the other posts and such, today we're going to be talking about tearless onions. So, for anyone who's ever cut an onion, you'll know that, as soon as you cut into them, you immediately start to cry. Well, I do. I seem to be particularly sensitive to this, and it's very frustrating because then you're just trying to have to, cook blind. And so, I've gotten very good at chopping onions without looking at them, and mostly through tears. But that's super annoying. So if we can, fix that, that would be good. Now, you might be worried that tearless onions means flavorless onions, and this is not the case. The thing that makes you cry, which is called a lacrimator, is not the thing that gives it its taste, although they are related. So we'll get into that. Before we really get into it, some announcements, as always, same sort of announcements. If you have not already, be sure to head over. We're talking about food today, and onions. So be sure to head over to my second channel, The Taste Emporium, for all of the food goodness that you could ever want. Of course, the bar here, this is the donation bar. If you donate through the link in the comments, it will go to filling up this bar. Donations through Super Chat and that sort of thing do not. We are currently fundraising to build a new editing computer. Because as I'm currently editing the new video, 90% of what I'm doing is waiting for my fucking computer to load. So, yeah, that's super exciting. So the point of the new computer is to fix that and generally speed up my ability to edit. It should mean nicer videos, more ability to do animations and custom graphics and all this kind of stuff. So that'll be really cool. If you want to donate, it's greatly appreciated. Really helps me out. And other donations through Super Chat and stuff go towards food and rent, both of which I'm told are important. But other than that, let's just get right into it. So, let me just... Mr. Roberts, thank you. Thanks for kicking things off right off at the beginning. Greatly appreciate it. Okay, let's get into this. So, because the topic is pre-chosen, I've already taken some time to do a little bit of research, so I'm going to just kind of go over it just quickly, because we had a question about the computer. Will you be going with AMD or Intel? I'm going with AMD. Getting an AMD Ryzen 9 3900X, I think. Going off memory here. But yes, I'm going AMD. Okay. So, I have already pulled up some resources to hopefully speed things along, and also just so I can kind of give a little bit of a prelude instead of, spending half an hour staring at research when I have no idea what I'm doing. So, first off, let's talk about propanethyl S-oxide. I'm going to mess up the name of this about 1,050 times, so just roll with it. This is the thing that makes you cry. It's the lacrimatory agent. Lacrimatory means makes you cry. So this compound is a gas, actually. The reason that when you cut into an onion that it makes you cry, when you actually cut into onion cells, there's an enzymatic reaction, which we'll talk about in a second, where a different compound is... So basically, there's the disarmed and the armed version of this compound. This is the armed version, also known as the angry version, also known as the version that makes you cry. When you cut into an onion, it triggers the release of the enzyme, or rather, it's sort of like mixing, to steal a line from Brad from Bon Appetit. It's sort of like a two-part epoxy, like a two-part glue. When the compound, the disarmed compound and the enzyme mix, they immediately react very, very, very quickly, and this stuff is produced. This is then immediately released as a gas, and then, fumigates the kitchen, and then you're just, left there crying and unable to see what you're doing. This is obviously a defense against things that want to eat the onions, because, unsurprisingly, this is unpleasant for most creatures. But humans are simultaneously stupid and smart, so we've realized that the crying is not actually dangerous, and so we kind of just, like, suck it up, and then we get to eat delicious onions. And I don't know about you, I love me some onions. So yeah, that's this. This is the, so what we've got to do is basically turn off the enzyme that arms this compound, and that should make the tearless onions. So let's talk about that enzyme. That enzyme is part of a family of enzymes called alanases. So there's all sorts of different alanases. This is one of them, and you can see that this is sort of the one of the precursors. This is allicin. So this is the thing that Brad is constantly talking about when he talks about medical grade garlic, which I love his videos, but it's the stupidest fucking thing that he says. That aside, this is the compound that he's talking about. It's called allicin. It comes from alliin, which actually is a derivative of cysteine. Cysteine being one of the essential amino acids. This stuff is in everything. Like, so many different proteins use cysteine. It's very, very important biologically. The way that you go from cysteine to allicin is you first stick this little dangly bit on here, and then the allanase nips the ends off and turns it into allicin. However, if you knock the little dangly bit off, you're left with the propanethyl S oxide, or something very similar to it. You'll see they're not quite the same. So this one has a double bond. So they're in the same family of compounds. This is obviously the one in garlic. The one in onions is different. But this is how you know they're all in the same family. They all use these weird sulfur compounds. So yeah, that's the that's the enzyme. The specific enzyme we'll look at in a second. But is this purely theoretical? Like, is this something that we've, just pulled out of a hat? No. This is actually something that's been done many times. So, allow me to introduce Sunions. Now, obviously this is not sponsored. This is just, I'm deeply entertained by the existence of this thing. But this is a variety of onions, which somebody has made to be tearless. These are a thing that you can get, especially in the States. You might have to look around a little bit, but they are actually available. So, Sunions, I believe, were not made through... Oh, somebody kicked in. Thank you, Katja. So, Sunions were not made through genetic engineering. They were just made through traditional breeding. And, because, I mean, the thing with traditional breeding is if you just mutate the plants, statistically, one of the mutants is going to have the gene, like the enzyme that does the conversion, broken. You'll probably break a whole bunch of other things, and that could screw with the onion, change its taste, change its other properties. But the important thing is that it doesn't make you cry, and then you can breed that back in with onions that are more flavorful. So this is described as the first tearless and sweet onion. So yeah, if anybody has Sunions growing near you, enjoy, because they sound wonderful. Also, I love the name. It's just cute, and their marketing is just adorable. So I just appreciate this. This is just cool shit. However, before Sunions, there was this paper. So there's actually several papers. This is just one of them. But for those who feel like following along, the name of the paper is Silencing Onion Lacrimatory Factor Synthase Causes Significant Change in Sulfur Secondary Metabolite Profile. So this is one of the ones that looks at what happens when you actually turn that enzyme off. And one of the things that they found was that it does actually mess with a little bit of the flavor compound stuff, because it's like a lot of enzymes are not one enzyme does one reaction. That's the thing they'll tell you in Bio 101, and it's a lie. A lot of enzymes actually have multi-function, like they'll act on many different compounds. So there is the possibility that it can mess with the flavor a little bit, although it's not necessarily the case. So, but I mean, with the nice thing with breeding is that once you've got this thing that turns this one enzyme off, then you can, mix and match with other plants to try and shift the flavor profile to compensate for any losses from turning this one enzyme off. So there was another paper, which I think this was the first one. This is the one I was thinking of. No, this is the same one. Never mind. Okay, so here's some of the sulfur compounds that they're talking about, and sulfur compounds, especially in biology, get super weird, like super weird, because I mean sulfur is sort of a fascinating atom, especially in the context of biology, because it allows for what are called disulfide bridges. It allows sulfur is sort of a very flexible compound. So it allows you to stick things together in interesting ways. It also bends back on itself in really tight angles, and so that ends up with really interesting compounds. But it's also very easy chemically to break these bonds. So this allows you to very quickly release compounds or break things apart in interesting ways. But also they can be very volatile and very irritating. So for something like onions, they're kind of the compound of choice for a lacrimatory compound. Cool. Now we know this. So this paper is called "Enzymes That Make You Cry: Crystal Structure of Lacrimatory Factor Synthase from Allium cepa." Allium cepa is the name of this is just the scientific name of onions, and over the years, this enzyme, because I mean, it's well known that this is the enzyme that arms the propanethyl S oxide. So it's been studied, and so this is one paper that's studying it, specifically looking at the crystal structure of it. And what's really interesting is the crystal structure data from here ended up on this site called Uniprot. So Uniprot is a website that we have used many times in almost every single live stream that I've done. And it's basically a protein database. So all enzymes are proteins, and so they are often in Uniprot. So basically what I did to figure this out was I basically looked up Tearless Onions NCBI. NCBI is it's this website. It's this massive database of like genes and proteins and papers and all kinds of stuff. So you this is sort of the trick. When you want to find some bio thing, you just go thing that you want NCBI, and then this should pop up. And so I just looked up like Tearless Onions, this, and they talked about it somewhere in the abstract. And because I was looking for the name of this enzyme, because when we were talking about the enzymes before, we were talking about alanases. Alanase is a very broad term. There are literally hundreds of them, at least 1 to 20 or 30 individual enzymes in every species of thing in the onion garlic family. So there's a lot of them. So you got to find the right one. More importantly, we need the one from Allium cepa, because if we want to make tearless onions, it wouldn't make sense to try and turn off a gene that isn't there. And especially when we're going to be trying to turn off a gene in a specific species, we need that specific species DNA code, or the silencing won't work. And so we're going to be doing the same thing that we did for decaf coffee, which is why I have this pulled up, and we'll come back to this in a second. But to do that, we needed the actual gene sequence. So I went to Uniprot. Now we can see what the enzyme looks like, which is very cool. This is the actual sequence. So we could actually we could do this two ways. We can either turn the enzyme off, or if you wanted and were a monster, you could actually upregulate this gene and add an extra copy of it to the plants, and make them extra teary onions for people who are bastards. But it is possible. I saw a couple of comments go by. Somebody said that they just read Walk Away and they really enjoyed it. So I'm glad you did. If you haven't, it's a fantastic book. That's Walk Away by Cory Doctorow. Highly recommend. Somebody asked, can you make a video about how to protoplast fusion? Yes, just as soon as I figure out how to actually get it to work. It's something I've been tinkering with for a while, but I'm getting there. So yeah, that's the thing. Okay, so then I went to this site, which is the European Nucleotide Archive, which I'd never heard of before. And this, okay, so what this is showing, this is a chunk of DNA. This is saying that it's from Allium cepa, so this is the Allium cepa genome. And then there's LFS, which is lacrimatory factor synthase, or lacrimator. Is it lacrimatory? Is it lacrimator? Yeah, lacrimatory factor synthase, LFS. So this is the actual DNA code, like the actual gene for this thing, which is what we want. Now, technically, we'd probably want the RNA for this, but because I had a look at this. So if we go to the sequence, you can see it's not very long. It is really, really quite short. So the chance of there being weird intron, exon shite in this gene are quite low because this is so small. That's generally something that you see happening with a really, really large gene, and also that tends to happen more in like mammals and animals. Plants, their genomes can be so freaking large that they just don't even bother. Like if they need 4 versions of one gene, they're just like, yeah, fuck it, just have 4. So, so thank you, Real Fight Frog, for donating. How about an extra layery onion for Shrek fans with superior onion smell? I don't know how you would do that. It sounds funny though. Okay. So anyways, we have the gene sequence here, so I'm going to just copy it. Copy. Now, the nice thing is we're just doing gene silencing, so that should be nice and easy. Anyway, so I've got the decaf coffee pulled up. First things first, so this website that I'm on right now is called Benchling. It's benchling.com. And it is basically a visual editor for messing with DNA. We've used it many times before. If you're unfamiliar with it, I would highly suggest checking out the earlier streams, since we have made basically every single one of these folders has been a construct that we have made on previous streams. So we've made a lot of stuff at this point. One of the reasons I really love doing these streams is because we get to just make cool shit. And like most of it is not something I'm ever going to actually have made, but just like going through the motions and designing it out has been a lot of fun. So this one we're going to call Tearless Onions. It's always good to make lots of folders and, non-destructive edits. I talk about this a lot, non-destructive edits. If you're going to change something, make sure you've got a copy of the original, because if you change it and it doesn't let you change it back, you're going to be very sad. So first things first, we're going to be reusing the decaf coffee thingamajig. So now I just got to stick it in the right spot. Okay, Tearless Onions, cool. So we now have this plasmid, and again, this circle here is a ring of DNA. It's called a plasmid. I've talked about this before as well. Where did I put Tearless Onions? Here we go. And so now there should be a copy. Yay! Copy of decaf coffee. Great. So while that's in there, let's also... Nope, that is not what I wanted. DNA sequence. LFS gene. Okay, so I'm just pasting in the LFS gene that we just got off of that other site, just so that we have it. Ooh, this is very interesting. All kinds of interesting restriction sites. Okay, so what we're going to do is let us review the decaf coffee and how this works. So This is the decaf coffee plasmid, and so in the same way that this plasmid works, wherein if you were to put it into coffee plants, it would make decaf coffee, like it would break the caffeine making gene. We're going to do the same thing, except in this case we're going to turn off the lacrimator gene. So there's a few different things in this plasmid which make it helpful. There are various genes that are basically there so that you can select for the modified plant, because that's really important. Like you end up doing a lot of this stuff in cell culture, and then once you've modified the couple of plant cells, you grow it out into a whole plant. And so a lot of the overwhelming majority of the genes here are for that. This I don't remember what it does, but it's probably not important. But this is the actual important bit that we care about. So I'm going to just click on it. So first things first. This is the Cauliflower Mosaic virus promoter. So this is a piece of DNA taken out of Cauliflower Mosaic virus, which normally is a bastard, but like all good things in science, we've taken a thing that's awful and made it not shite, or used it for our own purposes, is, I feel like, a more accurate description. So basically what this thing is really good at is just cranking out whatever comes after it. So if you've got, if you've got this, whatever gene you stick after this, you're going to get a lot of, which is good in a lot of cases, not so good in others. This, though, is the actual thing that we're going to be designing. It's what's called a small interfering RNA, and it's designed as a hairpin. So there's in the same way that a hairpin is, two legs and a loopy in the middle. It's exactly the same thing, and the two legs match. So this is the actual targeting section, right here, and it's not very long. It's 18 base pairs long, so it's really a tiny little piece. And so what this would do is this would actually stick to the RNA of the, in this case, the caffeine synthase gene, and it basically clogs up the system. So ribosome. Okay, so just as a quick review, this is a ribosome. This is the thing that actually makes proteins. So it's actually a cluster of proteins itself. But the idea is you've got this big chunky boy that's made of two main halves, the large subunit and the small subunit. The RNA, so when you've got a bit of DNA, right, just like this, the first thing that happens is this gets turned into RNA, specifically from right about where this arrow starts. So, the ribosome binds to, or not the ribosome, sorry, RNA synthase, RNA polymerase, binds to the cauliflower mosaic virus promoter, and then everything that comes after it, it turns into RNA. So once you've got that chunk of RNA, generally, and of course this is not the case with RNA interference like what we're doing, but say this was like the lacrimator gene, right? Like the lacrimatory synthase gene. That RNA is single-stranded, so the little teeth on the bottom are exposed. So the ribosome binds to it and then reads out the codons, so like 3 letters at a time, and then based on what 3 letters are there, and this is actually a better image. So you'll see that it's reading 3 letters at a time. And based on which 3 letters are there, a different carrier will stick and then transfer the amino acid that's stuck to it onto the growing protein chain, and that's how you get that. So the way RNA interference works is you basically have something that is the exact complement of the thing that's going through the ribosome. So what ribosomes require single-stranded DNA, or RNA rather. So if there's a thing stuck to it, it literally cannot run. It will clog up the mechanism and the ribosome will just let go. And more to the point, double-stranded RNA tends to get cut. So various other enzymes in the cell will actually recognize double-stranded DNA, or RNA rather, and cut it and destroy it. So it's kind of twofold. So A, it clogs the mechanism, and B, it forces that RNA to get degraded very quickly. So it's a very good way of silencing a gene without actually messing with the core gene. So like everyone, a lot of people would be like, oh yeah, just use CRISPR to break the gene, when in reality it's much easier to just do this, and this is actually way more specific. Yeah. So that's the thing. Okay, so moving on. Now we have to actually design the thingamadoober. So, as we said, it's 18 letters long. So we're going to need to find an 18, and we're literally going to do exactly the same thing. First things first, we're going to find, we're just going to pick 18 letters from the LFS gene, and that's going to be the front of the hairpin. Then we're going to use this same random bit of shmoo in the middle for the loop section, because it doesn't matter. It gets cut off and is literally irrelevant. And then we're just going to have the mirror of whatever we put here over here to finish off the hairpin. So let's do that. So let's go over to the LFS gene. I'm going to take a bit from close-ish to the middle, but we're going to avoid things like this, where it's just a string of A's, because that's not helpful and also makes targeting really hard. So I want to try and find something that is not too far from the beginning, but also not super repetitive. Something that looks nice and what should we call it? Like it looks. We want it to look nice, So I'm gonna just first things first. I want to just have a little look. See, okay, that's not right. So I'm trying to figure out where the start of the actual gene is. Because I suspect that this was not made properly. Okay, the bottom one is the fucked one, so we're going to get rid of that. Sorry, I'm just cleaning this up really quick. No, stop codon, no stop codon, no stop codon, stop codon. That's not good. Okay, this doesn't really matter what I'm doing. I'm just doing it because I'm curious and I want to try and figure out what the gene itself looks like. No, that's not right either. Weird. Yeah, we'll get it eventually. Oh no, that's 3. Very weird. Okay, so this seems like the best option here. Okay, well, that's weird. So yeah, I just wanted to see what the gene itself looked like, and considering that this goes most of the way down, how long was this on Uniprot? Because Uniprot said it wasn't overly long. 160, 170-ish. Yeah, okay, that tracks. Okay, so I'm just kind of talking out loud and thinking to myself. So the reason that I made this translation is for a few different reasons. When I looked at Uniprot, it said that the enzyme itself was only about 170 ish amino acids long. So I wanted to see, I bet you that's the start right here. So a couple of things. Enzymes always start with an ATG, which codes for an amino acid called methionine. So the reason that I made this translation was because I wanted to see where that methionine was. So now that we know that there's one here, and so I did this for 2 reasons. One, I wanted to see if there were any weird stop codons. So I'll show you what one looks like. So this is a stop codon. It's a little star here. This is generally marking the end of an enzyme, or the end of a protein. So the reason that I made this amino acid translation was to figure out where's the enzyme actually start, and also where does it end, because and if it's the right length, right? So if I now go from here to this first M, and I go create translation, this will all be the same, but now when we scroll down to the bottom, it should be about 170, excuse me, bit congested today. It should be about 170 amino acids long, and you can see that because this says 168, so this is 169, so it's about 170 long. Which means this is actually the right size. So we've probably found the like the entirety of the enzyme itself. Which means this is garbage. We can ignore this. This has nothing to do with the actual protein itself. And as such, we can ignore it, which is good. Why? Because if we hadn't done this check and we designed the RNA to like target right here, for example, it would have targeted nothing, and this wouldn't have worked. Cool. So now we know that. And this is also one of the reasons I checked, because I saw this, and this looks weird. You almost never see something like this or not never, but it's rare to see such a long stretch of just like A in a gene, unless it's at the end. So that was already setting off some alarm bells for me. I was like, I should check that. And sure enough, we've found the start. Looks like we found the end. So now we can actually choose a spot to target. So from what I remember from the decaf coffee, you wanted to go not too far in, but not too far down either. So I'm going to pick right about here for no particular reason other than it seems like a reasonable look. Actually, you know what? That's got that AAA in there, which I want to avoid. Let's go a little higher up. Okay, that's 18. So yeah, this looks pretty good. GC content about 52%. GC content generally makes it the higher the GC content, the higher the melting point of any particular piece of DNA. Which literally means that the two strands are stickier. So from what I remember about RNA interference, you want a kind of around 50% GC content for this to work really well. Cool. So the one thing I'm going to do really quickly before... Actually, I'm going to annotate this just so I don't have to figure this out again. Just label it as target. So that way I can just click on it, come back to it really quickly. I'm going to go back to the decaf coffee thing, and I want to see, because the caffeine synthase thing should be in here. So I just want to see... I didn't annotate it. Okay, that's annoying. But we can find that really quickly. So I'm going to go back to the decaf coffee plasmid, go over here, I'm just going to grab this, I'm going to copy it. I'm just trying to figure out where I targeted and which strand I need to target, because I assume, since we're making a thing that targets, that I need to be copying I need to be making the hairpin out of this bottom line of letters, not the top line, because the top line is going to be what the actual RNA is. So if we want something to stick to it, we obviously have to make its complement, which is the bottom line. But I just want to make sure that that's the case. Yeah, see? Okay, I was right. So this is the spot where we targeted last time, which I'm going to annotate, because always good to annotate your stuff. So you can see that when I pasted that into the search bar, the bottom line is highlighted, which, as I suspected, means we need to, for the thing that we're working on currently, the LFS one, we're going to need to copy the bottom line. Cool, no big deal. So we go back over the LFS gene, we click on target, we want to copy the reverse complement. So with Benchling, it gives you the option to copy all kinds of different stuff. So we don't want the DNA sequence, which is the top line. We want the reverse complement, which is the bottom. So if I just click it, that should copy it instantly. Now let's close the search bar because I don't need this. This is the copy of decaf coffee, so we're just going to go back to the Tearless Onions folder and rename this so we don't get confused. Rename. Somebody's asking, can we actually implement this into the real world? Yes, this has been done before. So there are multiple groups who have done exactly this. I showed some papers at the beginning where it was done. So yeah, no, it's very doable. Okay, so we're gonna highlight. Okay, I'm gonna click on that, and then it'll highlight it better. So this is, remember that this was the decaf coffee plasmid. Now we're making it Tearless Onions. So I just highlighted the targeting section from the last version of this. And by the way, if you want to know how we made this, go look at the previous live stream about decaf coffee, because we covered it there. So anyways, now that I've got the targeting section highlighted, I've already copied the correct thing from the lacrimatory factor synthase gene. So I should just be able to go paste. And you'll see that it changes. Cool. Now we just need to make this, we basically need its mirror to stick on the other end, and then we're basically done. This is kind of why I chose this topic, because this is actually quite easy. This would cost almost nothing to have made from like a synthesis company. So I'm going to just copy this. Oh, shit. I'm going to just highlight this again. I'm going to copy it normally. So this will have copied the front, or like the front, the top line. I'm then going to go back to the Tearless Onions. I'm going to grab the reverse part of the hairpin, and I'm going to paste it in. Now, the last thing that I got to do is just make sure that everything lines up properly, because you got to figure if this is going to loop back on itself, this this letter here needs to stick to this letter here, and so on and so forth. So I'm just kind of doing a quick little check. So if this is CTG, we'd expect it to be GAC, which is GAC, which it is. So then if it's ATA, we would expect it to be TAT, which it is. So this is good. These all line up properly. So now we're just going to fix our annotations, make sure everything's labeled properly. Reverse complement RNA I. We're going to make this a different color. We're going to make it blue. No, that thing's blue. I'm going to make it pink. Save. Then this is going to get changed to target. Actually, LFS target RNAi. Seek. And again, actually, you know what? I'm going to leave it yellow for literally no reason at all, other than I feel like it. Save. Okay. So we've got the front half of our hairpin, we've got our back half of our hairpin. This we didn't touch. This is literally a random string of DNA. It does not matter. In fact, just to show you what I'm talking about here, I just want to show you what this is going to look like. So this is basically what we've just designed. So we've designed the targeting section up here, the reverse section down here, and then there's this random bit of junk in the middle. This gets sheared off as soon as this is made, and then the two pieces pull apart. And then the top bit, which we care about, will actually go and stick to the RNA that it's targeting. Oh, Quinticle, thank you very much for donating. Greatly appreciated. So somebody has been asking about the milk and egg plasmids and have they been printed? I said in the last stream that yes, I've sent them into the, and here I'll bring these up just so you can have a look at them. So in the very first live stream that we did, we designed these two plasmids, although I made some small tweaks to them before I actually paid for them, because, you know, don't want to send in garbage. So the first one that we made was this one, which is the what I've been calling the Egg White Plasmid. Man, I'm really congested today. So this is the Egg White Plasmid. You'll see that it's got this thing that says ovalbumin. Ovalbumin being egg white. So this would be made in yeast, and the yeast would actually excrete this because of this little tag that we put on here. So it would basically, you'd have like a tank of sugar water, and it would slowly turn into egg whites, which is cool and basically modern alchemy, and I fucking love it. But yes, this has been submitted and paid for, and oh my god, was it expensive! But that was the point of doing the fundraiser, so that was good. It was just like, bio projects are so expensive. It ended up costing $12.50. So I think we did the fundraiser for $1,200. So it was basically bang on to what I expected it to be. So that was good. But so yeah, there were two plasmids that we sent in. The first one was this egg white one, and the second one makes milk. Uh, so we pulled the four proteins that are the main components of milk. In this case, we took them from deer, because deer was one of very few species that hadn't been patented. So this way, I can submit this final plasmid to a company called Adgene, and then anybody who wants it can play with deer milk, and there's no legal ramifications for doing so. So this is fantastic. This is the same with the egg white. So yeah, this way both will be made available once they're done. Like I've said in previous videos, though, making this DNA is it's a very slow process. This is a very complicated piece of DNA. So it's just going to take the company some time. It will probably take them anywhere from 4 to 6 months. That's just the speed of these things. Also, there is a fucking plague, and so they're kind of running a skeleton crew, so it's taking longer than usual. But it is getting made. It will be available. And once it's done, I'm going to put it into some yeast and then make a whole bunch of egg white and milk. And then make a cheese omelet. Because what is the point of being able to grow a cheese omelet if you can't make a cheese omelet? So yes, that will be lots of fun. And I'm still debating whether or not I want to sell the DNA myself, or if I'm just going to submit it to Adgene and make a point of saying that they need to sell it to whoever wants it. Because I don't have the time or facilities to be sending out, or like to be producing and sending out DNA like that. I'm still contemplating it. This might be, depending on interest, it might be something that I look into. Maybe I'll make it into a kit. I don't know. But if I put it up on Adgene, then it can just be immediately available for whoever wants it. And then if I feel like going the kit route down the line, or like partnering, there's a company called amino.bio, and there's a few other companies that sell DNA kits. So I might put together a kit and see if I can like partner with one of those companies and see if they'll sell the kit for me, and so that it can be made available. And that way you would get all the things you need. So you get like the yeast, you get the DNA, the transformation solution, all that kind of stuff. Yeah, that's something I'm working on. We will get there at some point. But yeah, like I said, we're waiting on the company to make the thing, and also once they're done making the thing, I then need to test it and make sure it all works properly. And that's a whole fucking thing in and of itself. I'd gotten one email that perhaps I'd made a mistake at one point, specifically that this sequence looked weird. Don't know. Like they said that this might be inverted. I don't actually know. We'll find out. If it is, there are these right here. These are restriction sites. If the way I designed this is to make it so that if any individual part of this plasmid is broken and doesn't work properly, I can remove it and replace it and fix it. So it is manageable. So Pop-Tart McJelly asked, so it's like open source genes. Yes, it is exactly open source genes. That is precisely what I'm doing. Speaking of which, all of the genes, although I'm kind of shit at remembering to do this, but all of the genes will eventually end up on this lovely GitHub page. This is the Whose Gene Is It Anyway GitHub repo, where I've put as many of the projects as I can. I'm going to be putting up more. The only reason there isn't the entire collection is because I am busy and forgetful. So eventually all of the things that I have in, or maybe not quite all, because there's this one, which we don't talk about, but all the ones that we made on the stream will eventually get on there just as soon as I remember to do so. So yeah. So right now, like, if you wanted any of the things that we designed on earlier streams, like say, I don't know, spicy tomato. Like if you wanted the spicy tomato construct, like you could download it, send it off to a company and be like, make me this, please. And they'll send you the DNA and then you can put it in a tomato and ta-da, you have spicy tomato. That's the idea anyway. I mean, it's bio. I assume most of this shit won't work, because that's just how it goes. No matter how well designed your construct is, the chances of it working on the first try, basically nil. So yeah, that's the thing. Okay, so since we're done, basically, I'm gonna take like 10 minutes, maybe 15, just to answer, because I'm seeing lots of questions in the comment section. So I'm gonna take a few minutes to just answer some questions, and then we're gonna wrap it up for the day, because I desperately need to get back to editing. So yeah. There was also a couple of donations which I missed, and so I want to just address them really quickly. Quinticle, who asks, quick question, if you have time to answer it, where would be the best place for one to start if he wanted to attempt to do something similar to what you're doing? If you have no idea what you're doing, I would always suggest the Manga Guide to Molecular Biology and the Manga Guide to Biochemistry. Manga Guide to Organic Chemistry is also good. Crash Course, Bio, Crash Course, OChem. Like, most biology is chem, so the more chem you can fit in your face hole, the better. But the molecular biology one is especially good because it's more talking about the both the biological systems and the way that you go about modifying stuff like this. In terms of actually getting started, so it's like, let's say you've taken the time to like build up that theoretical knowledge, the actual like brick and mortar, like get your hands dirty stuff. Your best bet is to pick up a kit. What? Dante says, I think you copied 19 letters, not 18. It's fine. The precise length doesn't really matter. As long as they're the same, then it's fine. Okay, as I was saying, so if you want to actually get started like doing stuff, then picking up a kit is really the best way to go, because it'll get you the most like hands-on experience, because then you can actually like do some modification, even though it's very paint by numbers and like your hand is being held the whole way through. Getting a feel for it is really, really important. But there are some great resources for this. The one that I mentioned briefly is this site, and again, this isn't sponsored. I mean, I'd love to partner with them at some point because I like their stuff. But these guys sell both a book and also hands-on kits and supplies and all kinds of stuff. And I really like their kits. They're one of the better designed kits that I've seen, because the only other ones I've seen are on Carolina, which I'll talk about in a second, and the Odin, which is shite. But I mean, they also have different ones for different skill levels. So like this is your like bare bones basic one. Oh, thank you, Nico. So these ones I like because they're colorful, and I am a sucker for colors. So these are, you genetically modify some E. coli to be colorful, but then they've also got ones that are a little more involved, which I really like. Where's the one that I was thinking of? The Smell It kit is quite fun. So this one is actually one that we've talked about on this stream, and so this one I really like because it's like the colorful protein ones are, I mean, they're cute, but this is I find this to be a more like viscerally interesting kit. So it encodes the ATF1 gene, which is acetyltransferase 1, and you basically mix isoamyl alcohol in with your culture, and because of the enzyme that you've engineered the bacteria to produce, it'll stick an acetyl group onto it and turn isoamyl alcohol into isoamyl acetate, which smells like bananas. So it's this really cool way of showing that like you can get flavor compounds out of genetic engineering. So kits like this are a really great start. There's another website called Carolina. They also have some kits. The one that I would... Oh, wow. Okay, that's very broken. Let's try that again, Carolina. If you'd be so kind as to not shit the bed. Thank you. So they've got a series called BioBuilder, which I highly recommend. because when they actually remember to send all the fucking reagents, um, they're really really neat. This is the one that I would really suggest. It's a little pricey, admittedly, um, but it's also meant for like an entire class. So if you've got some friends who want to like get in on this, you can all pitch in and kind of get this really interesting kit. So if I click on this, these are regular yeast, and so the video that I'm working on right now is this basically. It's not this kit. It's slightly different, but I do talk about this kit. Um, so it basically takes regular looking yeast and make them express beta carotene. So they turn vibrant orange, which it's not cooperating for some reason. But yeah, also a great kit, very hands on, highly recommend. Somebody said that they'll donate every time I say play with deer milk. So play with deer milk, play with deer milk, play with deer milk, play with deer milk. Okay, let's see if they actually donate. That'll be fun. Okay. Jonathan asks, how would one put the gene into the tomato plant? That's sort of a complicated question. There's all sorts of different ways to modify plants. The typical way to do this is with something called agrobacterium, which is basically you modify bacteria, which when you expose the plant to that bacteria, it puts the gene into the plant. The other option is a gene gun, which are easy to make and look dope. And they're exactly what they sound like. You literally shoot the genes into the thing. And like literally it can be like you take a piece of leaf and you just like pop and just like spray genes into it. So the way this works is you coat little gold or tungsten particles, like really tiny, like a couple micron wide particles or less usually, in the DNA that you want, and then you literally shoot it into the plant. And so typically you do this to like callus culture, which I'll just show you what that looks like. Plant callus. Callus, I can spell. So this is a little plant booger looking thing. So this is basically you take a piece of leaf or other plant tissue and you grow it on hormone plates, and it devolves into this plant booger that is basically a chunk of stem cells. You then shoot this with the gene gun with particles coated in the DNA that you're interested in, and then once it's had time to start expressing those genes, you transfer the little plant booger onto another plate that has the same hormones, but also an antibiotic that'll kill all of the plant cells that have not taken up the new DNA. And at that point, everything that's not modified dies off, everything that is modified survives, and then you let it grow into a whole plant by changing the hormone concentrations, and you end up with an entire plant that's been modified. This is also, doing it this way, where you gene gun a callus, is the only method that is allowed for release for food. Like if you intend on releasing a new crop, that's the only way you're allowed to do it. The agrobacterium method, even though it's faster, more efficient, works better, you're not allowed to sell the end product on the off chance, even though it's fucking zero. On the off chance that the bacteria is still present on the plant, because it could spread and that could be bad, and it's fucking stupid. It never happens, but legislation makes it so that you can only do it the hard way. Okay, Dante asked, can GMO things reproduce? Yes. Unless you've made them to not, then yes, almost all of them can. The gold nanoparticles coated in DNA. This was an older method, right? No, I mean it's older-ish, but like it's also pervasive and the only method you're allowed to use for anything commercial. Well, the only method you're allowed to use for modern genetic modification, which is fucking stupid. Like you're allowed to expose plants to just a cesium pill for days until they mutate and get all fucking weird. Or just an X-ray source, or chemical mutagens, or all kinds of different things, and that doesn't count as GMO for some stupid reason. You're also allowed to do protoplast fusion and all kinds of other things. Just when it comes to like modifying one gene at a time, the gold particles and gene gun is the only thing you're allowed to use. Do you know where to get kits in Europe? Doing genetic engineering in Europe. How fun. Yeah, that's not a thing. It's super illegal to do genetic engineering in Europe. It's one of the reasons why most genetic engineering happens in everywhere else. Um, like, even just putting GFP into an E. coli requires a fucking license in Europe, because Europe is backwards as shit in a lot of instances, uh, especially anything that has to do with technology. It's just the way it is. Supervirus has been donating. Thank you very much. Um, the play with deer milk thing, I don't get it, but play with deer milk. Hopefully people continue to donate when I say play with deer milk. Um, yeah, so that's the thing. Yeah, this is the reason why my European friend who loves genetic engineering, basically the way that he handles this is he'll design a plasmid. So it's like he'll come on, go away. So he'll make something like this and then have like a synthesis company make it, and then he'll send it to like me or my friend Gabriel or whoever else, and then one of us will actually do the genetic modification and like run the protocol for him, and so that he can still do genetic modification just remotely, and then you don't have to deal with Europe being Europe. Um, the stupid thing with, well, generally people who are anti-GMO, generally, is that all the research shows that GMOs are safe. All of it. There's absolutely nothing that shows that GMOs are any way harmful or worse. Most of the time, they're more healthy, or have been made to produce more food. They're totally safe. Frankly, they're safer than things made with the mutagen method, but the mutagen method is not regulated for some fucking reason. But Europe is backwards as shit and run by people who are approximately 1,000,000 years old and, frankly, like, dumb. Like, so, small rant there, but as a rule, legislators in Europe, dumb as shit. It's just the way it is. So yeah, it makes it very difficult to do anything there, which is why I'm not in Europe. Um, Supervirus, thank you for continuing to donate. I greatly appreciate it. I mean, we're slowly but surely moving that donation bar. Got a long way to go yet till computer time, but every donation helps. Oh, my first ban of the day. Come on. Hey, fucking cooperate, will you? There we go. Bye-bye. Get fucked. Yeah, I like when people are like, you're spreading propaganda. You mean things based on actual research and not, like, baby's first fraudulent research paper? Yeah. Propaganda. Also, that's the other thing. Like, people, especially when people are like, we need to go back to nature and natural ways, and they forget that, like, every crop they've ever eaten is just as man-made as every fucking Pop-Tart. Like, you think plants that you get at the grocery store were like that naturally? No! That's 10,000 years worth of selective engineering. Like, wait, actually, fuck it, I'm gonna pull this up because it's entertaining. I find this endlessly entertaining. Anytime somebody is like, oh, come on. I think ancient, maybe? That's not spelled right, but I don't care. What? Plants. Okay, I mean, here's a pretty good example. but maybe like. Okay, so this was from a painting, because it's one of the only references we have to what watermelon looked like before we bred them to be the monstrosities that they are now. Don't get me wrong, they're delicious, but this is from several hundred years ago. This is what a watermelon looked like. You'll see it's mostly pith, like, and rind. Like, there was almost nothing edible about it, but we bred it over thousands of years into this. Like, this is okay. So for reference, old watermelon, modern watermelon. You can see that there is essentially nothing the same. So yeah, yeah, this is the same with every crop that you've ever enjoyed, be it corn, corn versus. I mean, corn is a great example. This is. Yeah, here you go. Is that going to get bigger? Or is that just going to be? No. Okay, that's just going to stay small. Okay, so this is ancient corn on the left here. You'll notice that it looks nothing like what you think corn looks like. This is modern corn. This is 10,000 years worth of breeding. So yeah, this is yeah, it's a thing. It's the same with carrots, the same with eggplant, the same with literally every fucking plant you've ever eaten. Every plant available in the grocery store is just as man-made as a Pop-Tart. So anyone is like, GMOs are bad. I'm like, why do you hate puppies? like, fuck off. Somebody asked, what will the final computer be used for? Editing, making videos. As much as I love doing live streams, I also like making normal videos, and that's mostly what I do. That'll be the rest of my day. And currently editing is a slow, tedious process because my computer is old and laggy, and it's just a nightmare to work with. So the new computer will be going towards fixing that and building something that'll actually be sustainable and like useful, and it'll speed up my editing so that I hopefully can get videos out faster. Because right now a huge part of the editing process is waiting for the fucking thing to render or load, or just it crashes and then I've got to wait for it to reboot. It takes ages. Very frustrating. So that's what the thing goes for. Okay, we've got a bunch of requests for blue raspberries. So, okay, cool. Well, I'll throw it up on the next poll. Next week, on the next Who's Gene, we're going to come back to the magnetoreception thing, because I said I was going to finish that off today, but I really need to go edit, so I'm not. We're going to do, so next week we're going to do magnetoreception, and then the week after I'll put up a poll, and we'll pick a new topic. And one of them will be blue raspberries. I'm today super virus keeping it going. Awesome new basts. Would egg yeast make alcohol? Yes, it's still yeast. We'll still make alcohol. So that'll be a thing. Cosmic Mutant asks, Thursday food video. No. I'm working on a new video for the main channel. I'm trying to get it out before the end of the month. Well, not trying. I will get it out before the end of the month. And I can show you what it's about. This is the construct. This is literally the exact construct that I have, and I'll be showing how to make it. So these are regular yeast that have been modified to produce beta carotene. So it expresses this whole pathway, and I've done it. And so I'm going to be showing how I did it, how the pathway works, talking about what you can do with the yeast. I've also got, so I made these yeast, and now I've got a sourdough starter made with these yeast, so that the bread that you make would be full of carotene, so vitamin A. So yeah, that's the thing. Supervirus, thank you. Yeah, somebody was Supervirus the one who said this? Someone was like, "I'll keep donating every single time you say play with deer milk," and so Supervirus has just been kicking in. So thank you very much, greatly appreciate it. Okay, what else we got? On the topic of fungus that eats radiation, I've got a thing for that. Um, a friend of mine has made a plasmid that works in E. coli and plants, that produces melanin, and then there's this really cool paper that melanin producing bacteria or fungi or whatever grow better when exposed to radiation, and so it kind of does like radiosynthesis, which is neat, and so we'll be playing with that at some point. Yeah, somebody says making alcohol is illegal in Europe. Everything is illegal in Europe. Fun is illegal in Europe. Sanity is illegal in Europe. Yeah, fuck Europe. I like not Europeans. There's a lot of amazing people in Europe, but like European law is dumb. Um, okay, what else we got? The glowing plants thing have been half synthesized. Two of the genes got made properly. Two of them, the company kind of shit the bed and fucked it up. So that's getting reattempted, and if that company can't do it, then it's going to be printed by a different company. So it's making progress, but it's slow. Do you think eggs should be sold washed or not washed? I think it doesn't really make a difference, and washed eggs, when kept cold, last much longer. I mean, you gotta keep them cold, but they do last much, much longer, so technically I like the washed eggs. I mean, I like the idea of being able to keep eggs out on the counter. That's kind of neat. But then you gotta, like, do the float test and shit, make sure they're still good. Whereas washed eggs, you can keep in the fridge for frickin' ages, and they just last. Is golden rice banned in Europe? Probably. I mean, getting it deployed has been extremely difficult because people, like, burn the fields because they're dumb. Fucking Greenpeace drives me fucking crazy. Like, they're just so stupid. Like, they're, like, anti-nuclear and anti-GMO, and I'm like, literally those are the two things that could actually, like, fix the planet and, like, do a lot of good, and they're against it, and it's so stupid. Anyway. I'm not gonna spend too much time bitching about that because I could literally bitch about that all day. Okay. With that, I'm going to have to wrap it up because I really do need to go get editing, and I have a bazillion other things to do today. This plasmid will be uploaded to the GitHub soonish, as soon as I remember to do so. Thank you everybody who's donated. It really is greatly appreciated. Hopefully we can hit the goal soon so that I can get that computer built. That would be awesome. Oh, we just had okay, one quick question. How to remove Supervirus, thank you very much. How to remove beta carotene from crude capsaicin extraction. You could do chromatography, probably. Be my best bet. But I don't know. I do some chemistry, but I am not a chemist. But anyways, this has been a lot of fun. I'm glad we got to cover this topic finally. It was highly requested. The next stream will probably be either Friday afternoon or Saturday. I haven't decided. It's going to depend when I get my editing done. Because if I need more time, I'm going to probably be editing on Friday as well. This is a monster of a video. It's like 22 minutes long, so it's going to take me some extra time. But yeah, next stream will either be Friday or Saturday, and that'll be another biohack chat. Not sure who the guest will be yet. It might be Gabe, might be my friend Kazima. Not sure yet. We will figure it out. Thanks again for everyone who donated. There's links in the description to other bits of content. Yeah, that's basically it. I hope you guys are staying safe and having a good day. And yeah, just keep doing that, and I will see you on the next one. Thank you very much. Bye-bye.